Okay, while Will is getting ready, I'll go ahead and introduce him for his talk. He is an MCB major, also getting a minor in family ministry, and he's on the track team. Many of you may not know that. He's going to stick around one more year after this year to continue throwing the javelin, which is to me kind of cool. I like to go see that happen sometime. And because probably last year's eligibility year, so he can stick around and throw for another year. Intends to go to med school and intends to apply for some sort of a military program to help cover the cost of med school and then serve his country after that. So I respect that as well. He's going to be talking today about some of the effects of amphetamine and dopamine concentrations in the body and some of the mechanisms involved in how that occurs. So go ahead, Will.
Can you hear me? Okay. Let's see here. So today I'm going to be looking at how amphetamine is able to impact neuronal signaling by acting on dopamine transporters DAT and VMAT2. So animals with nervous systems use neuronal signaling as a way to interpret and respond to stimuli, whether that be, for example, a coyote in the woods if he were to catch the scent of a dead rabbit nearby or a larger predator in the area. He would then rely on neuronal signaling to interpret these stimuli and choose the appropriate response, whether that be to scavenge the dead rabbit or flee the threat of the predator. So in a situation like I just described, neuronal signaling takes place where stimulus is perceived by neurons and relayed as an impulse to the brain where it can be interpreted, and upon completion of the cognitive process, a response will be chosen and it will then be sent back out through neurons as needed. And in order for neurons to function in this way, they rely heavily upon a chemical messenger referred to as neurotransmitters. Neurotransmitters are stored in presynaptic vesicles where they await release that will come from depolarization in a nerve impulse that will trigger vesicle docking and subsequent release of neurotransmitters. And they will then cross the synaptic cleft where they will bind postsynaptic receptors on adjacent neurons and thus depolarize those neurons, passing on the signal. And today we're going to be looking specifically at the neurotransmitter dopamine. So why look at dopamine? Well, as we've kind of discussed in some of the presentations earlier this semester, dopamine has a largely known role for functioning in feelings of reward and pleasure. However, dopamine is involved in a lot more than just that. There are 4 pathways in the brain that rely on dopamine signaling, and these pathways collectively are involved in functions like cognition, working memory, decision-making, motor planning, metabolism, and inhibition of hormones like prolactin. So if an individual was to experience issues in their dopaminergic signaling, it could greatly affect their ability to function in any of these areas. And the issue we're going to be looking at specifically today is inadequate dopamine levels in the synapse. So this problem can arise typically due to one of 2 things, which are a decrease in release where the vesicles are not releasing enough dopamine into the synapse, or it could also arise from an increase in premature reuptake where the neuron is taking back in the dopamine after it's released it before proper signaling has taken place. And you may be wondering, like, how could these causes come about? Like, what would cause these issues? Well, there's many different variables that factor into signaling pathways, but there have been 3 common factors linked with dopaminergic issues, and these are disease, drug abuse, and oxidative stress, which is linked to aging. So what's the solution to this problem? Well, the best way to fix it would be to attack it at the source and increase release and decrease reuptake. And researchers found a possible solution to this problem in the form of amphetamine. Amphetamine is a synthetic stimulant drug that has a monoamine structure very similar to that of dopamine, and it's slightly basic in nature. So our first question, we need to determine the legitimacy of amphetamine as an option. So we need to look at whether or not it can actually increase extracellular concentrations of dopamine. So the first experiment we're going to be looking at, done by Zestos et al., uses an in vivo technique called microdialysis. And this technique, as described by Zapata et al., involves the use of a probe inserted in the brain of test subjects, and this probe has a dialysis membrane, which allows diffusion of neurotransmitters such as dopamine into the probe. And then it also has an inlet and outlet tube connected to the probe. This allows perfusion of liquid, and in the case of our experiment, it was artificial cerebral spinal fluid. And when they perfuse this liquid in and out, they're able to collect samples from the probe that can later be tested to determine the quantification or the levels of neurotransmitters that were in the tissue at that time. So in this experiment, they had male Sprague Dawley rats that were fitted with these microdialysis probes, and then they were housed individually with many different factors kept constant, such as light and food and things of that nature. And these rats were then treated with an intraperitoneal injection of amphetamine. And after this injection, they used microdialysis to record the changes in concentration. In this study, they looked at a lot of different neurotransmitters, but we're going to be focusing solely on dopamine today for the purpose of our talk. And so if you look here, you can see the x-axis, you have time in minutes, and y-axis, you have units of percent that are relative to a baseline recording that was established prior to treatment. They established the basal levels of dopamine release, and everything else on this graph is a percentage relative to that. So at 0 here, you can see they administered the treatment of amphetamine, and there was a large increase in dopamine concentrations after treatment. And this increase was found to be statistically significant through one-way ANOVA, or repeated measures one-way ANOVA. And these findings are also backed by another study done by Davercall et al. And in this study, they did another in vivo technique, but this one is fast-scan cyclic voltammetry. And basically what this experimental technique is they insert a carbon fiber microelectrode into the brains of these rats, and then they apply voltage to the electrode. And this voltage causes the oxidation and reduction of dopamine molecules. And first it will oxidize them, so as you can see, the hydrogens will leave the hydroxyl groups, and then subsequently they will return after the voltage wave returns to a ground state. And this movement of electrons back and forth creates an alternating current. And this alternating current can be used to gather inferences about the change in concentrations of dopamine in real time. So in this experiment, they fitted the male Sprague Dawley rats with this voltammetry probe, and they then administered intraperitoneal injections of either a control group, which was saline, or the treatment group, which was varying levels of amphetamine. And if we look at this first graph here, this is a depiction of dopamine release into the synapse. So on the x-axis, you have time in minutes, and on the y-axis, you have units of percent relative to a basal level that was determined prior to administration of treatment. So they, at 0 here, you have the treatment being administered, and as you can see, there is a significant increase in the dopamine release in these neurons of the amphetamine groups. But the control group, which were just given saline, there was no significant change at all. And these findings were found to be statistically significant through repeated measures one-way ANOVA. In the next graph we're going to look at from this same experiment, they were also able to record the reuptake of dopamine in these dopaminergic neurons. And so this graph here, you have time again, minutes, and on the y-axis you have units of percent relative to the basal level of reuptake. So as you can see here at 0, once the amphetamine treatment was administered, the treatment groups experienced a large decrease in reuptake, meaning that they were taking less dopamine back into the cell after it had been released compared to the control group, which had no significant change. And these changes were found to be statistically significant through a repeated measures one-way ANOVA. So to kind of summarize what we've learned off the bat on our first question, we've been able to see that amphetamine does have the possibility to increase dopamine concentrations, and it's able to do this by increasing release and decreasing reuptake. So this leads us to our next question, which is how is amphetamine able to do this? What mechanisms of action does it have, and what is it doing to increase its release and decrease its reuptake? So before I kind of throw you into the deep end, I'll try and give you a little background as to what we're going to be looking at, which is two transporters, the first one being dopamine transporter, or DAT, and this is a plasma membrane transporter responsible for taking amphetamine back into the presynaptic neuron after it's been released into the synapse. And the next transporter we're looking at is vesicular monoamine transporter 2, or VMAT2, and this is a transporter on the vesicle membranes responsible for storing dopamine in the vesicles to later be released. So the first study, we're looking at the function of DAT and amphetamine actions. And this study, they use microdialysis again. Microdialysis, like we saw before in the first experiment. And the main difference in this experiment was they were treating mice, and there were two different types of mice that they were treating. They had a wild type group with a normal functioning dopamine transporter. So there's going to be regular reuptake occurring. And they had a knockout group, which did not have a functioning dopamine transporter. And they were looking at the differences between these two to see how essential the dopamine transporter was to amphetamine's actions. As you can see in this graph here, the wild-type group exhibited the response you would typically see with the y-axis representing dopamine concentrations relative to basal level that was established prior to treatment, like in the before graphs, and minutes on the x-axis. So you can see this expected response here, but in the knockout group, you do not see that response, and there's no significant change there. And this finding supports the current proposed methods of action of amphetamine in the dopaminergic neurons. And this proposed mechanism is, firstly, amphetamine will compete with dopamine to be taken into the cell through the dopamine transporter DAT. And after that competitive inhibition, amphetamine enters the cell, and it is believed to trigger the phosphorylation of DAT. And once DAT has been phosphorylated, the conformation changes from an inward conformation to an outward conformation, where it is now functioning in efflux of dopamine rather than reuptake of dopamine. And this change in conformation wouldn't increase the dopamine release in a typical sense, because dopamine is mostly concentrated in the vesicles of these presynaptic neurons and not in the cytosol. But this is where VMAT2 comes into play. So VMAT2, as we said before, is a vesicular transporter, and VMAT2 is responsible for storing dopamine in the vesicles, and it depends on a proton or pH gradient to accomplish this. And so we're going to be looking at the effects of amphetamine on this pH gradient. So in the next experiment done by Freiberg et al., they used a transgenic line of Drosophila flies, and they used whole brain samples from these flies. And these flies had a biosensor called d-VMAT-fluorin inserted into their sequence, which basically added a fluorescent tag to the VMAT transporter. So whenever the pH of these flies, the vesicle pH in these flies increased, the biosensor would fluoresce. So they took these transgenic flies and they decapitated them and removed their brains for testing in BAF superfusion. And there were several different tests they ran. They had control groups, which consisted of a chloroquine treatment, and this chloroquine treatment was used because they knew chloroquine could diffuse across the plasma membrane and the vesicular membrane independently of VMAT and cause alkalization of the vesicles. So this served to show what a strong response would look like. And they had another control group, which was the vehicle group, which was just the bath superfusion in the medium in which the amphetamine would be administered in. So there should be no change in this. So this chloroquine group represented a maximum response, and the vehicle group represented a minimum or no response. And then there were varying levels of amphetamine that were treated as well. So if we look at this first image here, you can see this is a Drosophila brain that is both before and after the treatment with amphetamine PEP. And beforehand, you can see very low levels of fluorescence, which indicate the normal pH in the vesicles of the neurons here. And after treatment with amphetamine, you can see a strong increase in the pH of these vesicles, as indicated by the intensity of the fluorescence. And these results are quantified on this graph here. And as you can see, starting at 1 micromolar concentration, amphetamine was able to elicit a significant increase in vesicle pH. And this was determined to be statistically significant by way of one-way ANOVA. And they also did another test with repeating the same procedures with a VMAT inhibitor in the bath. And so by doing this, they were able to prove, or at least provide some evidence that VMAT plays a vital role in this vesicle alkalization, because with inhibited VMAT, the same effect was not seen at all. And to kind of give you a recap of what we learned from these last few studies of the mechanisms of action, firstly, amphetamine is able to reduce reuptake of dopamine, and secondly, it's able to increase release. And it does this through first competing with dopamine to enter the cell, and then once it is entered, it also causes the subsequent phosphorylation of the DAT transporter. And this reverses it from that inward to outward conformation. And then another function of amphetamine once in the cell is the alkalization of neuronal vesicles. And when these vesicles are alkalized, it causes VMAT2 to efflux dopamine from the vesicles rather than to store it. And once this dopamine has been released into the cytosol, the newly reversed transporter, the newly reversed DAT transporters are there to then release this readily available dopamine. And all of these functions, in conclusion, work together to increase your dopamine concentrations in the synapse. So, in conclusion, amphetamine enters the cell by way of DAT, and it also alkalizes vesicles of presynaptic neurons by way of VMAT2. And these two things function together to increase the dopamine levels in the synapse and also delay the reuptake of dopamine from the synapse. Thanks for listening.
Thanks, Will. Interesting talk. We'll turn it over to Dr. Pittman for questions from the chat.
Thank you, Will. I'm going to try to combine a couple questions here about uptake, reuptake versus release. And kind of one part of that is, how do they determine that the problem is not enough release or too much reuptake? And going along with that, how do they measure that reuptake?
So is the question you're talking about in regards to not during experimentation, just like in normal life, if they were able to, how they could measure efflux versus reuptake?
Yeah, go with that.
Okay. Well, I didn't see anywhere in my research that they're able to actually measure this in more of, I guess you'd say, a clinical setting. They are able to indirectly measure dopamine levels through a technique I saw where they can, doctors can get images of the density of dopamine transporters in the brain, and they can use this to infer about dopamine concentrations. But I don't think there's any way for doctors or researchers to determine whether it's reduced efflux or premature reuptake that's causing the problem. I guess apart from them performing microdialysis or fast scan cyclic voltammetry, and I don't think those would exactly qualify as non-invasive procedures.
So on slide 15, how did they measure the dopamine uptake?
On slide 15, how they measure the reuptake. So in the fast scan cyclic voltammetry, they emit a triangular waveform that starts around a voltage of -4 or -0.4 to up to, usually it's 1.2 is the standard, and then it returns back to that -0.04 or 0.4. And while the voltage is increasing, that's when oxidation occurs. So as the dopamine that is being released, the hydrogens will leave these hydroxyl groups. But as the voltage wave returns down to the ground state, that's when the reduction occurs. And so these produce two different, there's another type of figure that I didn't have time to go over because it's kind of a lot, but they have the current versus the voltage rather than the what we see here with the, this is what they interpreted from that graph. So they're able to see a current that is due to release as the oxidation occurs, and then there's a subsequent current farther down the line that is due to the reduction as the dopamine molecules are being taken back in by the cell. So it's just two different currents that they have, and the oxidation current allows them to measure release, and the reduction current allows them to measure the reuptake.
Thank you. And a question from Dr. Rule, slide 14. Did the researchers test for an interaction between amphetamine concentration and time? Based on your understanding of the data, do you think they could have?
Do you mean like how amphetamines affect like how long it takes them to wear off, like the half-life, or do you mean between these two groups how you see they act differently over time?
I believe the interaction between the concentration and the time. So how
yes.
each...
Okay. Well, they found that they did a one-way ANOVA testing the significance between the two different concentrations of amphetamine, and they found that to be significant. They did a ANOVA on that as well, and that was this same probability here, a probability of less than 0.05. And so there was a significant difference between the response between the two groups, and they I think they hypothesized that there's a less intense response in the 1 milligram group. However, it was a more sustained response, and I think this was just due to here you're releasing a lot more of the available dopamine, so it's all out there, and then from this point on, it's all just reuptake. But as you can see here, it's still continuing to release while the 10 milligram group is going down. So it doesn't reach its maximum amount of dopamine release until much later compared to the other groups. So this results in a more sustained level of dopamine in the synapse, if that's what you're asking.
I don't see Dr. Rule making any motion, so I think that is sufficient.
I don't think, is my mic working?
Yeah.
I can hear you now, yes, sir.
Oh, okay. Yeah, I was just wondering if they tested for the interaction specifically, like what you're talking about. Or if they could have, if you know if they could have.
Yes, sir, they did test, and they found that there was a significant difference between the two groups.
Okay, so question on slides around 24 to 25 on the Freiberg data. What is the change in F and change in F max measuring on the y-axis?
looking at here. So they used kind of a complex mathematical equation to get a quantification of the intensity, where they the chloroquine group that offered the max, they factored in that maximum concentration that was seen there, or maximum fluorescence seen there, and they also factored in the minimum or practically zero fluorescence seen in the other control group. And they were able to create a unit from that, and then they just compared change in fluorescence with a ratio to that I guess number or value that they determined from comparing those two. So basically, it's just a change in fluorescence related to the possible max and possible minimum.
Thank you, Will. And that is all the time we have for questions. But if you see Cassidy Waldron or Dr. Donnelly, they have questions for you too. So thank you very much to both Will and Caleb. Good job, guys. Congratulations on being done.